already. As Lionel was saying, I'm going to present today a simple fluorescence imaging technique that allows hyperspectral analysis of images that are acquired in highly scattering media. So you already have listened a lot about hyperspectral imaging, but I'm going to highlight some important points to show the method. So hyperspectral imaging has many applications, and is a technique that consists of measuring the emission spectrum in each pixel of the image. And at least you have a 3D data set that has two spatial dimensions, X, Y, and one spectral dimension with a wavelength. So the optical setup in the microscopes for hyperspectral imaging consists of having spectrometers in the microscope, and they work well, but they have the caveat that they are designed to work with collimated light. So they don't work well with scattering samples. So, and also are an expensive addition of the microscope. So at the LFD, we have been working in developing new methods that work well with scattering media. So also you have seen a lot about the spectral phasor approach, and Lorenzo just did an awesome presentation with this part, but how to analyze all the data, the massive data that we obtain with hyperspectral imaging is an easy way is doing the spectral phasor approach. And we transform the emission spectrum of every pixel of the image in one point in the spectral phasor plot. So in a summarized way, the spectral phasor approach is a mathematical transformation in where we transform the profile, the emission profile. Oops, the pointer goes. Well, out here. Okay. The emission profile we multiply by a cosine or sine functions, and we divide, we normalize by the total to obtain the GNS coordinates to plot in the spectral phasor plot. And as you already listened before, the position of these points in the phasor plot, they depend on the center of mass of the emission spectrum. For example, here, that has more blue emission, appears here in the phasor plot, but increasing in wavelength, they appear counterclockwise in the plot. And also the distance from the center, you already have seen that depends on the width of the emission spectrum. So the wider, the closer will appear. And also this property of the phasor approach, that is the linear addition property, that if we have two emitting species with different spectrums, with different maximums, and they are not mixed in one pixel, they will appear in different positions in the phasor plot. But if we have them mixed in the same pixel, that point will appear in a line that joins the combination of the individual species. So with this, in 2018, our group, Sasha, Enrico, and also Lionel, started developing a new method that instead of measuring the emission spectrum in each pixel of the image and then making the phasor plot transformation, the idea was to use transmission filters that you put inside your microscope, the optical path of your microscope, and you acquire images through those filters, and then you calculate with those intensity images the phasor plot coordinates. So you don't have the whole spectrum anymore, but you get information about you get spectral information. So what are these transmission filters with the sine and cosine profile? For example, here you can see, for example, in these particular ones that I'm showing, if you have this cosine filter, has maximum transmission at 400 nanometers, while the sine has half of that transmission and so on. So yeah, I'm going to eliminate this. Okay. Okay. So what we do is to take 3 images that are an image with acquired through a cosine filter, an image acquired through a filter with a sine transmission, and then we acquire it with total, like with no filter at all, to have the total intensity for normalization. And then with that, we calculate the phasor plot coordinates and plot in the spectral phasor plot. And you also heard before about this reciprocity principle that exists in between the plot and the image, where we can select ROIs in the phasor plot and see where those pixels are in the image and vice versa. So with this method, can be used with scattering media, and it's cheaper, these just filters, than the spectral detectors. But in this case, we need 3 measurements. So, or you need 3 detectors to do it at the same time, or you need to do it sequentially, that involves time. So now we propose a new method that is applying this sine and cosine approach, but is using ideal linear filters up and down. So means that the transmission in one of the filters increases while the wavelength increase, and the opposite for the other filter. And with this, we calculate a GP function where S and G are the intensity measured through these filters, and then we normalize by the total intensity, that is S plus G. And this GP function is proportional to the wavelength, and with this we can calculate the angular and spectral phasor position. So I'm not going to show results of this part because of time constraints, but we use commercial plastic filters that we bought on Amazon. And then also Sasha made these linear filters that was mixing dyes in plastic. And with these filters, we were able to separate different emitting species. So with this method, we simplify the acquisition to just 2 measurements. And but in this simplification, we have some losses. So the linear addition property that I have said before, it doesn't apply anymore, because now the modulation is fixed and we don't have sensitivity to the width of the spectrum. But this method is still useful to separate different spectra, and it could be applied to any monotonic filter up and down. Could be exponential. It doesn't have necessarily to be a linear one. So then we were like, but maybe we can simplify this even more, because the up and down filters are not independent, are related with the total intensity. So we said, well, we can actually image through a linear up filter and then the total intensity, and that ratio will be proportional to the wavelength, and then we can calculate the phasor plot coordinates. So it's a simple method, has been published this year in Biomedical Optical Express, and in the following slides I'm going to convince you, or try to, that this simple method is enough to have spectral information in your sample. So here we use our real linear filter up that is not an ideal filter, And this is, as I said, like a dye mixed in plastic. It was just that, the filter made by Sasha. And we tested it in our diver microscope that you saw before. We used to test it monochromatic light at 20 different bands in the range of 400 nanometers to 590. And as you can see here, all the different bands appear well separated in the phasor plot. You see as just a line that maybe looks different of what you have seen about spectral phasors, but remember I said that now we don't have sensitivity in the width of the spectrum and the modulation is fixed. That's why they appear in that way in the phasor plot. And then we tested it with a scatter light. So we added a piece of ground glass in the optical path, and we did the same experiment. And we, as you can see, the separation of the different light of different wavelengths is perfectly fine and well separated in the phasor plot. So then we use beads to test the scattering light also. So here we have beads of 2 different colors, blue and yellow-green beads. And in the first part, you can see with no scatter. And in the second one, we added the ground glass, and also they appear in the same positions in the phasor plot. So the scatter light is not a problem for this method, and we could prove that separates scatter light of different wavelengths perfectly well. So this method can be used with samples like tissues that are intrinsically scattering. So we also tested with Rhodamine solutions. We used Rhodamine 110 and Rhodamine B. And as you can see here, they appear in different positions in the phasor plot. And then we mix them, one-on-one mixture. And as you can see, appears in the middle of the angular position of the individual ones. So even though I mentioned before that we lost the property of linear addition, there is a relationship in the angular position. So with this, we wanted to test our method to study the cell metabolism that you yesterday heard talk about Suman Ranjit, about the NADH autofluorescence that allows to study metabolism in cells. So the NADH is an enzyme cofactor that is involved in metabolism and exists in 2 different forms. In the cell, it exists as a free form or bound to the enzymes, that is what we call protein-bound NADH. And has been seen that this ratio of free to protein-bound NADH can be correlated with cellular metabolism and has been extensively used using FLIM, because these 2 forms, they differ largely in lifetime. But as you can see here in this picture over here, the emission spectrum of NADH free or bound differs just slightly. So has not been used that much because they're very similar in their emission spectrum to differentiate these 2 different species. But we wanted to test if using our method, we could differentiate them using the spectral information. So first we tested solutions of free NADH and bound NADH to the LDH enzyme. And as you can see here with our linear up method, we see them in different positions at the phasor plot. And also the free NADH appears at longer wavelengths, as has been previously reported, as expected. And we compared that result with a spectral detector of the 880 microscope, and we could see the same, that free NADH appears at longer wavelengths, and we could separate them. So then we tested our method in cells. So these are NIH-33 cells, are live, and we excited them with 740 nanometers, and this is a representative image of like autofluorescence image, and was collected in the range of 400 to 500 nanometers to avoid the FID contribution. And as you can see here, we have, well, the phasor plot, and we color coded this distribution, this phasor distribution with this scale that you can see here. And in the image, in the color coded image, you can see the majority of the pixels with more contribution of free NADH appear in the nucleus. And the bound NADH appears in the cytoplasm, in the mitochondria, as expected. And we compare, since in the diver we have the capability to do FLIM at the same time, we compare with FLIM, that is the method that everybody uses for this purpose. And you can see in the image that has the same pattern. And we also compare with the performance of the commercial spectral detector of the 880 size microscope. And again, we can see the same. So with this, we could tell that this method can be used to study metabolic changes in living samples, can be give information that is complementary with the FLIM information if you have in your microscope that capability. If not, you can use just the spectral part, and that gives the opportunity of people that cannot measure FLIM that can use these filters to study metabolism. And on top of that, it's good to use with tissue samples that are scattered samples that can give you problems if you try to do it in other setups. And now we're trying to improve our system because, as I mentioned before, we need a consecutive, we have in the diver detector, we have just one detector, so we need to do consecutive measurements. And when you have live cells, they move, the mitochondria moves. So to improve that, we are installing a multi-channel detector. In our case, we have 4 channels. And you can see here that we can put different filters. In this case, we have sine and cosine filters. This is the total intensity with no filter. Here we have our linear one, but we can combine and put all difference of whatever we want, and also to measure FLIM at the same time, and we can put like an NADH filter, etc. And the other thing is like we need that from the excitation point, all the photons arrive to this. It's a wide area detector, and then we need the photons to travel to the different channels. So we needed to add a scatter plate inside, so the photons could travel and reach the different channels in the detector. And well, we're working on that, so that's it. So as a conclusion is that this technique can be applied to any fluorescence microscope equipped with a single detector or camera. It's exceptionally good with scattering samples, as I said, as biological tissues. And it's simple and an inexpensive method, and the acquisition can be done in a single measurement if you have a multi-channel detector. And